Hello everyone, welcome to Molecura. Today we're going to be talking about regulation of glycolysis, and we're going to go through them one by one and see which molecules help in regulating glycolysis in the cell. We have already talked about glycolysis and how it happens in the previous video. Now, here we're going to be talking about the regulation of glycolysis. So, we are mainly concerned about three steps in glycolysis. The first one is going to be the conversion of glucose to glucose 6-phosphate, and the enzyme that's responsible for this we have talked about is called hexokinase. In the liver it's called glucokinase. Hexokinase is regulated by its product through feedback inhibition. So glucose 6-phosphate comes and inhibits hexokinase. It's going to be different for glucokinase, which is in the liver, because glycolysis is going to be more important to continue in the liver even in the presence of enough glucose 6-phosphate. So it is going to be regulated through gene expression by insulin, for example. Now the second one is going to be Phosphofructokinase 1. This is the rate limiting enzyme of glycolysis, which means that most of the regulation that's happening in glycolysis is usually happening through phosphofructokinase. And lastly, the last step, which we have phosphoenol pyruvate getting converted to pyruvate. This is happening through the action of pyruvate kinase, and this enzyme is also highly regulated in the cell. We will be looking at the first reaction now in greater detail. So this reaction that happened, we already talked about it as the first step of glycolysis. We had our glucose, which was getting converted to glucose 6-phosphate. And this step is happening through the action of hexokinase. And we talked about it, that hexokinase is actually taking off one phosphate from ATP and it's putting it on our glucose, thereby using or utilizing ATP in this step. Here we are at our phosphate group, which was going to be on carbon number 6, and that's why the name is glucose 6-phosphate. Now what happens in glycolysis? This step is going to be regulated by glucose 6-phosphate itself. So when this reaction is happening a lot, we are going to have a lot of glucose 6-phosphate, and what happens is that glucose 6-phosphate comes and tells hexokinase that we already have enough. You can go ahead and you can stop this reaction from going any further. So this is an example of feedback inhibition. Feedback inhibition. So hexokinase is inhibited by its product, which is glucose 6-phosphate. One more thing that can happen here is going to be by the action of insulin. So what happens is that we have our cell here. This cell has a nucleus. This is our nucleus. And in our nucleus we have our DNA. So what happens? Insulin comes and tells the DNA that we need more hexokinase. So insulin is telling the DNA that we need more hexokinase. So the DNA gets transcribed into mRNA, and this mRNA is translated into hexokinase. So insulin is going to indirectly affect the gene expression, thereby increasing the amount of hexokinase in the cell. So this happens when we have a lot of glucose in the body. So the action of insulin is to stimulate the gene responsible for producing hexokinase, thereby increasing the mRNA that is responsible for making hexokinase, and translation of this mRNA into a protein is going to be producing our hexokinase. So this in a nutshell is the first step of regulation of our glycolysis. So now we come to our second reaction that is regulated, and that is our phosphofructokinase 1. If you remember from the video on glycolysis, this step is converting fructose 6-phosphate into fructose 1,6-bisphosphate, and this step is also utilizing ATP to put the phosphate group from ATP on phosphate number 1. This enzyme therefore is going to have two active sites. The first active site is going to be the reactant, which is fructose 6-phosphate, and the second one is going to be ATP. But this enzyme is also having 4 allosteric sites, which we will talk about in details. Here we have our fructose 6-phosphate, and our fructose 6-phosphate is getting converted into fructose 1,6-bisphosphate, and this is happening through the action of PFK. And let's try to draw our PFK here. This is our enzyme, which is called PFK. PFK1 to be exact. Now, this enzyme is going to have 2 active sites, as we talked about. The active sites on this enzyme is going to be here and is going to be here. So the first active site is going to be occupied by fructose 6-phosphate, and the second one is going to be occupied by ATP. And as we said in the video about ATP, is that ATP is almost always attached to magnesium or any cation with 2+ charge. So these are the 2 active sites of phosphofructokinase. And as we said, here we are going to have also ATP getting consumed into ADP. And as we talked about, here on carbon number 6 we had a phosphate group, but here we are going to have 2 phosphate group on carbon number 6 and on carbon number 1. Now what about the allosteric sites that we talked about? PFK1 is going to have 4 allosteric sites. What are allosteric sites? We have talked about it in the video on enzymes, which you can find it on the biochemistry playlist. Basically, allosteric sites are sites that bind to certain molecules, and binding of those molecules is either going to activate or inhibit that enzyme by either increasing the speed of the reaction or by changing the enzyme in a way to make it more readily available to get connected to the substrate on its active site. And here our substrate is going to be fructose 6-phosphate and is going to be ATP. Now, there are 4 allosteric sites as we talked about. So let's try to talk about them one by one. First, let's talk about what is going to inhibit phosphofructokinase 1. Our PFK1 is going to be inhibited by 2 things. First one is going to be ATP. Of course, when we have a lot of energy in our body, ATP levels are going to be high, and this ATP is going to come to our phosphofructokinase and it's going to be inhibiting its action. So ATP is the first one that is going to inhibit our phosphofructokinase. The second one that's going to be inhibiting our phosphofructokinase is going to be citrate. Now citrate is an intermediate in TCA cycle. So when TCA cycle is happening a lot, we're going to have a lot of citrate laying around. So that's why citrate is going to be telling phosphofructokinase, enough. We have enough ATP. You can shut it down now. We don't need glycolysis to go on. So now we're going to be talking about what is going to enhance or activate the action of phosphofructokinase. The first one is going to be AMP. So the first one that we're going to be drawing here is going to be AMP, or adenosine monophosphate. Adenosine monophosphate is going to come and is going to activate our PFK. Now, why AMP and why not ADP? The point is that the level of AMP in the cell is going to be more reflective of the energy state of the cell. The reason is that the amount of ATP in the cell is a lot, okay, followed by the amount of ADP and followed by the amount of AMP. So a small change in ATP, it's going to reduce the percentage by a small amount. Let's say we have 100 ATP in the cell. If 2 get removed, now the amount of ATP in the cell is at 98%. However, AMP's concentration in the cell might be 10. If we reduce this by 2, we are going to have 8, and that's going to reduce it from 100% to 80%. So you see that the changes in the amount of AMP in the cell is going to be more reflective of the energy state of the cell than ATP or ADP. Furthermore, we have a reaction in the cell that is going to synthesize ATP from ADP. And this reaction goes like this. We are going to have 2 ADP getting converted into ATP plus AMP. And this happens through the action of an enzyme that's called adenylate kinase. So adenylate kinase is going to be converting our ADP into ATP and AMP. Therefore, the decrease in ATP is going to be less sensitive than increase in AMP in telling us how much energy does the cell have. So this is going to be an important thing to remember. Let's remove this for now. Now let's talk about the third one, which is going to be more important than the rest of them and is a bit confusing. It's activation by fructose 2,6-bisphosphate. So we have something here that we call fructose 2,6-bisphosphate. And fructose 2,6-bisphosphate is also going to be activating our PFK1. Now, how does this happen? This is going to be an important topic to discuss. So in the liver and some other tissues, we have an enzyme called Phosphofructokinase 2. Here you can see what we have is PFK1, which is Phosphofructokinase 1. But Phosphofructokinase 2 has an action to activate PFK1. How does that happen? Now, let's try to write this down here. This is our fructose 1,6-bisphosphate. Sometimes our fructose 6-phosphate could undergo action of another enzyme that is called PFK. So this is our fructose 6-phosphate. So what happens is that this fructose 6-phosphate could undergo action of another enzyme that is called PFK2, phosphofructokinase 2. And phosphofructokinase 2 can make another molecule which is called fructose 2,6-bisphosphate. Now, as you can see, the cell is trying to bypass the inhibition by ATP and by citrate. Why? Because in the liver, some reaction needs to go on and it does not need to stop despite having high energy levels. So fructose 2,6-bisphosphate is going to be a potent activator of glycolysis that comes here and is going to activate PFK1, which is phosphofructokinase 1, to tell the cell to keep glycolysis active. Now, PFK2 by itself is going to be regulated as well. PFK2 usually has two domains, a kinase domain and it also has a phosphatase domain. So this is going to be happening in both ways. Now, of course, here also we are having an ATP getting used and it's getting converted to ADP. The kinase domain is the one that is making fructose 2,6 bisphosphate. Why? Because it's taking a phosphate on ADP and is putting it here on our fructose 6 phosphate. So the way this molecule looks like is going to have a phosphate group on carbon number 2. That's why it's called fructose 2,6 bisphosphate. It also can have a phosphatase domain, which is going to remove this phosphate group and convert it back into fructose 6 phosphate. And this is going to reduce the amount of fructose 2,6 bisphosphate from here, and it's going to remove this activation, and therefore citrate and ATP are going to win and are going to stop PFK from going forward. So PFK2 by itself is getting highly regulated. How? It gets regulated by the action of insulin and glucagon. So in a state where glucose is widely available, insulin is going to increase the action of phosphofructokinase 2. Why? Because it needs glycolysis to go on. So in a fed state, insulin is going to come and is going to increase the action of phosphofructokinase 2 by dephosphorylating it. Conversely, in a state of starvation, the liver needs to do gluconeogenesis. And gluconeogenesis is going to be important to make glucose for the rest of the body. So glycolysis should not happen in this situation. That's why glucagon is going to come and is going to phosphorylate phosphofructokinase 2. And this phosphorylation is going to be inhibiting phosphofructokinase 2. What happens next? This does not get produced and there is no activation of PFK1. And again, citrate and ATP are going to win. So let's try to summarize this one more time because it could be confusing. In our third step of glycolysis, fructose 6 phosphate was getting converted into fructose 1,6 bisphosphate. Here, phosphofructokinase 1 was the enzyme which was making this reaction possible, and it was using 1 ATP to take the phosphate of this ATP and put it on carbon number 1 of fructose, thereby making fructose 1,6 bisphosphate. Now, PFK1 is going to have two active sites. One of them, of course, is for the substrate, which is fructose 6 phosphate. And the other one is for ATP because it's using ATP to turn this substrate into the product. These are the two active sites of phosphofructokinase. Now, phosphofructokinase is highly regulated. It has 4 allosteric sites. When the energy level is very high, ATP is going to be high, and therefore ATP has an allosteric site on phosphofructokinase which is going to inhibit it. At the meantime, citrate, an intermediate of the TCA cycle, is going to be increased when energy level is high. So it can come and tell phosphofructokinase to please stop glycolysis. When ATP is getting used and is getting depleted, it's going to be converted into AMP, which is adenosine monophosphate. And adenosine monophosphate level will increase when ATP is decreasing. Therefore, it's only logical that AMP is going to be activating phosphofructokinase to tell the enzyme that we need more glycolysis to happen because we need energy. The last one, which is the confusing bit, is fructose 2,6 bisphosphate. Fructose 2,6 bisphosphate is going to be made through the action of another enzyme, which is called PFK2. Now, what happens in some tissues like the liver? We need glycolysis to go on even in states where energy is high. So what happens? Our fructose 6 phosphate through the action of PFK2 is going to be converted into fructose 2,6 bisphosphate. So what happens is that our fructose 2,6 bisphosphate is getting made to come here and keep stimulating our PFK1 and to tell this enzyme that glycolysis needs to happen non-stop. So we are bypassing the inhibition that is going to be put through the high energy levels by ATP and by citrate. PFK2 by itself is also regulated by hormones, namely insulin and glucagon. Right after we have had a meal, the glucose level is high, insulin is going to come here and is going to dephosphorylate PFK2. And this dephosphorylation is going to tell PFK2 to please go ahead with this reaction. Why? Because we are going to need fructose 2,6 bisphosphate. Why? Because despite the energy level being high, we need PFK1 to work and to make more pyruvate. Conversely, when we are starving, someone who has not eaten for let's say 12 to 14 hours, glucagon level is going to increase. What will happen is that glucagon will come and phosphorylate PFK2, and it will tell PFK2 that please don't make fructose 2,6 bisphosphate. Why? Because we don't want PFK1 to work. Why we don't want PFK1 to work? Because we need to use the intermediates of glycolysis to make glucose. It needs to shift the biochemical pathways of liver into gluconeogenesis because the body is starving and we need to make glucose for the rest of the body. So glucagon will end up removing this strong activator of PFK1, so glycolysis can happen at a slower rate than when the body is well fed. Because PFK1 and its regulation is very important, I'm going to talk about it one more time on the main page on our main page of glycolysis. So here is the reaction of PFK1. It's converting fructose 6-phosphate into fructose 1,6 bisphosphate through taking one phosphate of ATP, converting it to ADP and putting this phosphate on carbon number 1. We said that our PFK1 is getting regulated by 4 things. So our PFK1 is here. This is our phosphofructokinase 1. So our phosphofructokinase 1 gets inhibited when there is a lot of ATP. So ATP is going to inhibit it. ATP inhibits phosphofructokinase 1. Now, when we have our pyruvate here getting converted into acetyl coenzyme A, we have our pyruvate through the action of pyruvate dehydrogenase gets converted into acetyl coenzyme A. And acetyl coenzyme A is going to go into TCA cycle. So our TCA cycle, it's starting by a molecule called oxaloacetate. We're going to talk about this more in the TCA cycle video. It's reacting with acetyl coenzyme A, and it's giving us a molecule we call citrate. So this is going to be our citrate. And now when we have enough citrate is going to come here and it's going to inhibit our phosphofructokinase 1. So this is our TCA cycle. And our intermediate in the TCA cycle, which is citrate, is going to be inhibiting phosphofructokinase 1. Furthermore, we could have activation of PFK1 by the action of AMP. AMP, when the energy level is down, ATP is getting hydrolyzed to AMP, so AMP is going to increase our phosphofructokinase. And also our fructose 6-phosphate can get converted through the action of PFK2 into fructose 2,6 bisphosphate. And this fructose 2,6 bisphosphate can come and cause activation of PFK1. So this is going to be what's happening with our PFK1. Now, the next one that we're going to talk about is going to be what happens here, and we will briefly talk about what happens here. In this reaction, our phosphoenol pyruvate is getting converted into pyruvate. And through this reaction, two molecules of ATP is getting generated. This reaction is happening through the action of pyruvate kinase, which is also highly regulated in glycolysis. So let's talk about that. So as we said here, we have our phosphoenol pyruvate. And our phosphoenol pyruvate is getting converted into pyruvate. This is our pyruvate. And this is happening through the action of an enzyme we call Pyruvate Kinase. And in this reaction, as we talked about it earlier, 2-ADP is getting converted into 2-ATP. Now, our pyruvate kinase is going to have different isoenzymes in different tissues. For example, the isoenzyme that is available in skeletal muscle is called the M1 type. This one is not going to get inhibited because in skeletal muscle there is little to no gluconeogenesis happening. Okay, so our phosphoenol pyruvate can get converted into pyruvate because this pyruvate is going to be needed to make ATP through TCA cycle and electron transport chain, or through anaerobic respiration, it's going to be used to produce more energy, especially during intense exercise. This is going to be the topic of our next video. However, the pyruvate kinase that is in other tissues, especially the liver isoenzyme, which is called type L, is going to be inhibited by influence of a couple of things. The first one is going to be phosphorylation through the influence of glucagon. So when we are at an starving level, we don't want our phosphoenol pyruvate to get converted into pyruvate. Why? Because we need our PEP for gluconeogenesis. So we don't want this to happen. The liver is responsible for providing glucose to the other cells in the body. So it will tell pyruvate kinase to stop. We are starving. We don't need to make more pyruvate. What we need to do is to take this phosphoenol pyruvate and shunt it to gluconeogenesis. So in this starvation, what is going to be the driving factor for this thing? And you have guessed it right, is going to be glucagon. So what happens is that glucagon is going to stop pyruvate kinase from moving forward, and this is going to be our glucagon. And how does glucagon do this? Glucagon is going to inhibit pyruvate kinase by phosphorylating it through CAMP dependent protein kinase. CAMP dependent protein kinase. Furthermore, there are some allosteric sites on pyruvate kinase. So pyruvate kinase also has allosteric sites of its own. And there are 4 things that I want you to remember. One of them is what activates it. What activates it is going to be fructose 1,6-bisphosphate. As you know, this is the product of PFK1 in glycolysis. So if PFK1 is working and is making a lot of fructose 1,6-bisphosphate, it means that we want this fructose 1,6-bisphosphate to get converted into pyruvate. Thereby, fructose 1,6-bisphosphate can come and also activate pyruvate kinase. Pyruvate kinase can get inhibited by a couple of things, and that is going to be in situations where energy level is high. And situations where energy level is high is going to be when we have a lot of ATP, and when we have a lot of alanine. So when we have a lot of alanine, it's going to indicate that the energy level of the body is high. So it's going to be stopping pyruvate kinase. We're going to be talking more about amino acid metabolism and catabolism in the coming videos. Now, there is one more thing that is going to inhibit pyruvate kinase, and to make that point, I'm going to be talking about pyruvate dehydrogenase. Now, we know that pyruvate, after it gets made in the cytosol through glycolysis, it's going to be converted into acetyl coenzyme A, which in the mitochondria is going to go through the TCA cycle, and it's going to end up producing a lot of ATP in the electron transport chain in the inner mitochondrial membrane. So for the pyruvate to get converted into acetyl coenzyme A, we have the action of an enzyme that's called PDH. So here we have our acetyl coenzyme A, and this is going to happen through the action of an enzyme called pyruvate dehydrogenase. Pyruvate dehydrogenase. When acetyl coenzyme is made enough, it's going to tell pyruvate kinase that please stop making more pyruvate, because we already have enough acetyl coenzyme A. So acetyl coenzyme A, in turn, it can come here and it can stop pyruvate kinase from making more pyruvate. So acetyl coenzyme A is also here working to inhibit pyruvate kinase. Now the question that we need to answer is that what is going to be regulating our pyruvate dehydrogenase? Now for that we need to answer this question, that what happens to pyruvate? The pyruvate can get converted into acetyl coenzyme A, it can go to the TCA cycle, and it can go to the electron transport chain. For this to happen, you are going to need oxygen. So in absence of oxygen, this acetyl coenzyme A is going to be useless. So the action of pyruvate dehydrogenase therefore is going to be useless. So in conditions when there is no oxygen, NADH is getting built up. So when the ratio of NADH is increasing, it's going to be something that stops pyruvate dehydrogenase. So here we can write it as when NADH ratio to NAD plus is increasing, it's going to be stopping pyruvate dehydrogenase from going ahead. This is important to understand, that when we have a lot of TCA cycle happen, we are going to be increasing the level of our NADH. But this NADH is not going to the electron transport chain, because the last acceptor of electron is oxygen. When oxygen is not there, NADH has nowhere to unload its electron. So its amount is going to increase while the amount of NAD plus is decreasing. So this increased ratio is going to be inhibiting our pyruvate dehydrogenase. Why? Because it's going to tell the cell to keep your pyruvate for anaerobic respiration, as we are not able to produce any ATP from oxidative phosphorylation because we don't have oxygen. So save it and shunt it back to anaerobic respiration. That is going to be the first thing. Now we will have to tie this to our phosphofructokinase 1. Here we have increased NADH. So it's going to stop PDH or pyruvate dehydrogenase from working. On the other hand, the energy level is going down. So this is going to increase our AMP, and our increased AMP is also increasing our glycolysis. And this means glycolysis will go on because it's going to need more pyruvate to convert to lactate, and for lactate to enable glycolysis from going on. We are not able to make our 30 to 32 ATP through electron transport chain, but we still need to make some ATP, although it's in a small amount, through glycolysis. So this is the reason that increased ratio is going to stop PDH, but increased AMP is going to make glycolysis happen over and over again. This is going to be the topic of our next video, and we are going to talk about anaerobic respiration in greater detail. Now, to make things a little more complicated, we have two enzymes that are going to be working on PDH to activate it or deactivate it. One of them is going to be called PDH kinase. PDH kinase is going to be phosphorylating PDH. It's going to be phosphorylating PDH and thereby inhibiting its action. So PDH kinase is going to inhibit PDH from going forward by phosphorylating it. On the other hand, we have something we call PDH phosphatase. PDH phosphatase, as the name suggests, is going to dephosphorylate PDH and it's going to activate it. During intense exercise, what happens is that the muscle is getting contracted over and over again. What happens during this muscle contraction is that there is going to be a release of a lot of calcium inside muscle cells. So we're going to have increased amount of calcium in the muscle cells. And this calcium is going to come and is going to increase the activity of our PDH phosphatase. So the PDH phosphatase action is going to be also modulated by intracellular calcium, because when we are exercising intensely, muscle is contracting, calcium is getting released. This calcium is going to come and is going to activate PDH phosphatase. PDH phosphatase is going to come remove this phosphate from PDH, thereby activating PDH, thereby activating more acetyl coenzyme A, thereby making more TCA cycle and more electron transport chain. So by this, we have covered the most important points that you need to know about glycolysis regulation in the cell. If this is helpful for you, please make sure to subscribe to our channel and like the videos. The playlist of our Medical Biochemistry Series is going to be linked in the description. You can find the videos in order on that playlist. Thank you for watching.